Good afternoon, everyone. Welcome to the H3D Foundation webinar series. Our today's speaker is Dr. Greg Basarab. Dr. Basarab is working as consultant for H3D and UCity. Dr. Basarab is highly capable, motivated, and well-renowned scientific leader with expertise in medicinal chemistry, infectious diseases, and drug discovery. He has experience in all phases of drug discovery, including early stage target validation and lead identification, lead optimization, preclinical evaluation, and early stage clinical trialing. He is the primary inventor of two drugs to enter human clinical trials, the inventor of over 30 patent applications, and author of over 50 publications in peer-reviewed journals. Today's topic for webinar is Adventures in Tackling Clinically Validated MTB Targets by Novel Means. Before we start with the webinar, the journal rules that we will have a Q&A session after the talk. So you can either post your questions in the chat or you can ask it at the end of the talk. Welcome, Dr. Basarab. Now I would like to request you to share your work with us. Over to you, Dr. Basarab.
Well, thank you very much for the very nice introduction. It's kind of exciting to be part of this H3D Foundation webinar series. I think it's very good to have this, and I'm glad that it's being carried out. Hopefully, there's no load shedding for the folks in South Africa, and otherwise we'll be able to get through it without a problem. So as advertised, I'll be talking about working on clinically validated MTB targets, coming up with new compounds for that. So let's get into this. I'd like to start with talking why MTB. I'm sure a lot of you already know this, but let me do the little introduction where what we all know is TB is still a problem in the world. And in fact, it's the world's leading infectious disease killer. More people die of TB than die of AIDS. And actually, more people now are dying than COVID-19. Certainly when COVID-19 had its spike, more people were dying of that. And it's in the heyday of the epidemic. Currently, as of the last reading in 2020, there are 1.5 million deaths due to TB. And this may have actually upticked a little bit higher, or maybe higher, during the COVID pandemic, not only because if somebody is compromised in lung function because they have TB and they get COVID co-infection, it's hard to say whether they're dying from TB or from COVID. A fairly good percentage of new infections are multi-drug resistance, and that's a big problem. It's really, really hard to treat. And South Africa has the largest per capita TB incidence and mortality in the world. And TB is largely much more problematic in the low and middle income countries. This is a problem because a lot of them can't afford the medicines that are needed. And I'll speak to that on the next slide. Here are some of the medicines that are used for treating TB. In the box, this is the most common first-line therapy. It's a combination therapy using 4 drugs: rifampicin, isoniazid, pyrazinamide, and ethambutol. But this takes upwards of 6 months to achieve a cure. And I want you to compare this. Suppose you have strep throat, which is caused by the organism Streptococcus pyogenes. You take a 5-day course of antibiotics, azithromycin is one that's commonly used, and you end up with a cure. Much, much harder to have compliance when you have a 6-month regimen of multiple drugs, all of which are going to have side effects. There are several second-line therapies. I'm showing that over here in the middle, that are used in various combinations for treating multi-drug resistant TB and extremely drug resistant TB. And these regimens can last for over a year. So TB is curable, but it's very, very difficult to cure. And you can imagine with these long regimens, it's hard to get patient compliance. That's our goal. Let's get a shorter, safer TB drug regimen. There are more recent drugs that have come onto the formulary or are in the pipeline in phase 3 clinical trials, and I show them there. Now, H3D has been involved for a long time in the TB Drug Accelerator Program. This is a Bill and Melinda Gates Foundation sponsored consortium. This involves multiple partners. At last count, and maybe this number has changed, there were 9 universities involved, 5 large pharmaceutical companies, 2 small biotechs, 6 research institutes. I guess that's where H3D comes in. Public development partnership outfit, and a philanthropic being Bill and Melinda Gates. And they work together pre-competitively to come up with new TB agents and position these agents for development. In development, you're going to need a new round of funding in order to figure out how to get compounds, in order to enable compounds to go to humans and eventually into the clinic. So the approach was taking compound libraries, a lot of these provided by the large pharmaceutical companies, carry out phenotypic screening, find hits, and carry on optimization programs to come up with compounds that could work in the body. There is work done for target deconvolution. We'd like to know the mode of action of these phenotypic screening hits. The kind of main goal most projects are going after is to get in vivo proof of concept. So it's not just getting good MTB activity. Obviously, it's also trying to get appropriate PK properties that it would work in an animal model. Alternatively, or the other approach is to identify targets. And there's a lot of work that's been done by the groups here in stratifying targets, validating targets, getting crystal structures of targets to be able to enable structure-based design, and screening the target to identify hits. And if you're good enough, you end up with hits of sufficient potency that you end out with whole cell activity in culture against MTB. You carry out the hit lead optimization program and eventually get to your in vivo proof of concept. All of this is directed to identifying preclinical candidates. So I'm going to talk kind of a hybrid of all this, and we'll call it target repurposing. You may have heard of drug repurposing, so I've put in this idea of target repurposing as taking scaffolds, identifying novel scaffolds that inhibit validated targets, and we want to inhibit these differently. As we know, in any given target, inhibitors can bind to the substrate binding site, the cofactor binding site, allosteric binding sites. They can disrupt monomer-monomer interfaces for those enzymes or proteins that have multiple units. They can inhibit different catalytic sites, and they can interact with metals differently. And I'll have an example of that within this talk. So all of this has to become prepackaged, or you have to be able to circumvent cross resistance, even though you're inhibiting the same validated target. And if you're able to do that, you've solved the issue of target validation. You know that this target may be valid because these targets have compounds that have been made it all the way through the clinic. They've solved particular issues associated with safety, that you can inhibit this target and come up with a safe enough drug to be able to use clinically. And hopefully we can shorten the drug discovery pipeline through this. Now here's a quintessential target. This is the ribosome. This is not necessarily for TB. This is for antibacterials in general, but there must be 50 to 20 ways, I don't know how you count it, to be able to inhibit translation, the conversion of RNA to protein by the ribosome. And here's, kind of the chemical entities and where they may inhibit and what state they may inhibit in the translation process through that. So lots of different ways to inhibit the ribosome, and this applies to other proteins as well. Probably not as robust as what you might see for the ribosome. I'm going to go into 3 vignettes. The first one of work that's been done at H3D, the first one being RNA polymerase. RNA polymerase is responsible for manufacturing RNA from template DNA. It's a relatively large protein. There's 2 large subunits, one 151 kilodalton, the other 155, some smaller subunits, and a sigma factor is required for activity. This is a target of rifampicin, which I had showed you on the previous slide, or previously. Rifampicin is a first-line TB agent. It works wonderfully well. It gets penetration into macrophages and has high concentrations in the epithelial lung fluid, where you want to be. You want your compound in the lung. It binds to the DNA RNA channel of RNAP. So that's shown here in a crystal structure that's been derived. The ball model is rifampicin. You can see DNA partially unwound bound, and in the background there's a little bit of RNA. So this is a structurally enabled project, and the problem with this is there's considerable resistance to rifampicin in the clinic, and obviously in particular in multi-drug resistant and extremely drug resistant TB. So H3D has entered into a collaboration with several other research outfits to look for other ways to inhibit RNA polymerases. And they formed a consortium within the context of the TBDA. So this consortium has put together an RNAP inhibition assay. They have a hypomorph assay. They have rifampicin resistant strains to work through, and they have an X-ray crystallography program to help support the project. I'm showing here some structures of switch region inhibitors, and switch region inhibitors bind differently over here, and there's kind of a part of the protein that opens and shuts that's allowing DNA and RNA in. So this binds differently than where rifampicin, and these compounds are not cross-resistant to rifampicin. They haven't been used or are not appropriate for using clinically for treatment of TB. Fidaxomicin is used clinically for the treatment of Clostridium difficile. So this is just an example of a series of inhibitors, and the team continues on trying to find new molecules that inhibit RNAP differently than rifampicin, and therefore not have cross-resistance. Next area I talk about is HAD-AB, HAD-BC, which is involved in mycolic acid biosynthesis. Here's the biosynthetic, if you will, circle that involves that's responsible for construction of mycolic acid. Mycolic acid is a long-chain fatty acid that's part and parcel of the cell wall of mycobacteria. So within this, there's a step in the in this sequence that is inhibited by isoniazid. Again, one of the frontline treatments for TB. This is a hydrogenation step that converts a double bond to a single bond and allows the cycle to carry on. Now, HAD A, B, and BC are responsible for the dehydration step. Early on, you have short chain fatty acids, if you will, that are run through this. HAD AB is primarily responsible for that. And as the chain gets longer and longer, HAD BC takes over. for the elongation process through that. Now, there are inhibitors that have been used clinically for HAD-AB, and these are thiocetazone and isoxazole, I've shown right here. So I mentioned this. These enzymes are important for mycolic acid biosynthesis, cell wall biosynthesis. These are actually, in a sense, kind of prodrugs. They're activated and form a covalent attachment to HAD-A cysteine 61. This is distal to the active site, and that's how they operate, these two drugs. If you knock out HAD-AB, they're deleterious to the pathogen. You cannot grow MTB, those knockouts. Knockout BC, the pathogen is still viable for growth. So HAD-AB can take over, if need be, for the activity provided by HAD-BC through that. So there's a collaboration between H3D and Colorado State University, Mary Jackson Lab, that was established based on a discovery at H3D from phenotypic screening of this compound, which was a screening hit that showed a pretty good MTB activity and was later showed to be an inhibitor of HAD-AB. Optimization work attained much better MICs, and eventually a crystal structure of an advanced compound was obtained with HAD-AB. This hit was discovered by phenotypic screening. It is not cross-resistant with other MTB agents, including thiocetazone and isoxazole. In trying to figure out its mode of action, it was shown to be involved in cell wall biosynthesis by a bioluminescence reporter assay that had been developed at the University of Cape Town. With this reporter assay, a light signal is seen when you disrupt cell wall biosynthesis in the engineered cell that was created. Transcriptional profiling also was shown that there is an upregulation of cell wall biosynthesis genes in response to these compounds, and that makes sense if you're struggling to get the components that you need in order to live, or the bug needs in order to live. It tries to do something that upregulates the genes involved in cell wall biosynthesis. Growing in culture in the presence of one of these compounds led to a spontaneous mutation in HAD-C. That was a K157R mutation. This mutant happened to have been known to compensate for HAD-AB inhibition. So the conclusion that ended up coming out that these compounds are actually good inhibitors of both HAD-AB and BC, but it's HAD-AB that's responsible for the bacterial kill. And if you generate this mutant that can compensate a little bit for HAD-AB inhibition, that maybe it keeps the bug viable. Pull down experiments showed binding to both HAD-AB and BC. Work done in the Mary Jackson lab showed that these compounds are potent inhibitors of both enzymes. But it's HAD-AB that is required for MTB viability and is responsible for the kill that comes from this class of compounds. So this project is ongoing. The main thing it needs to do is come up with compounds that have better pharmacokinetic properties in order to work in vivo. Oh yeah, and I had mentioned there's a co-crystal structure that was solved with these compounds. So it is a structurally enabled project. So the last area I'm going to talk about involves DNA gyrase. I'll spend quite a bit more time in this area on a project that we had at H3D. Now, during the DNA replication process, it's a complex process where DNA has to be unraveled, and each strand of DNA gets individually replicated. So several enzymes are involved in the replication process, but in order to unwind DNA and undo the tangles that you get in unwinding DNA, you need the enzyme DNA gyrase, and that operates before the replication fork, before these two strands are individually separated and individually copied. And it removes the torsional strains by inducing supercoiling in DNA during the replication process. There's another enzyme, topoisomerase 4, which is also called CE. I should have CE, not EC. That decatenates the DNA strands, the individual duplex DNA strands that are created during the replication process. So when these strands are replicated, they can kind of intertangle with one another. If you cut one, remove the other away from it, and then religate, you undo that tangling and let these individual DNA fragments, DNA strands, go their separate ways for that. And so these are two of many important enzymes that are essential for DNA replication. I had mentioned that DNA gyrase and topo4. In MTB, DNA gyrase carries out both the supercoiling activity, the release of the torsional strain before separating of the DNA strands, and the decatenation, the intertwining of the strands, and allows them to separate out. There is no topo4 through that. Now, topo4 and DNA gyrase are highly homologous enzymes. They operate by very similar mechanisms, and even in bacteria otherwise that have both topo4 and DNA gyrase, there is some crossover where topo4 can do some of the DNA gyrase activity to a certain extent. Now, I'll go over some of the DNA gyrase inhibitor classes that were already known, that are well known. The first are fluoroquinolones. Moxifloxacin is used for the treatment of TB. It's very valuable, and it shows excellent penetration of macrophages, and it does accumulate in lung fluid. A fairly recent clinical study showed that its combination with 3 other drugs, rifampicin, isoniazid, and pyrazinamide, reduced the time to cure to 4 months. So that's very encouraging. The problem with this is there's considerable resistance to moxifloxacin in the clinic. And in fact, I think fluoroquinolones were used before they were ever approved, were used for treatment of TB before they were ever approved for the treatment of TB. And they were probably used at suboptimal dosing, and that led a lot for that it is fertile ground for development of resistance. Another drug I'm showing here is novobiocin. It's an aminocoumarin natural product, antibiotic. This had a market presence for gram-positive infections. It really wasn't used that much, and it was discontinued in 2007. There's been a lot of work trying to get compounds that operate like in the same, if you will, mechanism that novobiocin does. Novobiocin is a competitor for the ATP binding site of DNA gyrase. There's upwards of 20 distinct chemical scaffolds that inhibit competitively for ATP on DNA gyrase and topoisomerase 4, I should add. many of which have MTB activity. However, none of these have progressed into the clinic as of yet. There is one drug that is now being clinical trialed. It's an ATP competitive inhibitor for non-mycobacterial for non-tuberculosis mycobacteria. This next class is this compound GSK 000. It's an naphthyridone class. This is potent in vitro activity against MTB. It has a distinct binding site from both fluoroquinolones and the ATP sites. As of yet, no analog, however, is progressed to the clinic for TB. A lot of this class in general suffers from issues of hERG inhibition and other toxicity issues, and it's been really, really tough to come up with something that is worthwhile. However, there is a drug, gepotidacin, that is in phase 3 clinical trials for gonorrhea. It's actually completed its phase 3 clinical trials for treatment of E. coli urinary tract infections. And it's on schedule for regulatory approval this year. So there's a compound in the class, but it doesn't show sufficient MTB activity. So it says that this class has promise in itself. Now, the area I'll talk about are an area called a spiropyrimidine trione, SPT, and there is a member of this class, zoliflodacin, that is currently in phase 3 clinical trials for the treatment of gonorrhea. So this is a DNA gyrase inhibitor and actually a topo-4 inhibitor too. But it doesn't show any cross-resistance to any other antibacterial, including fluoroquinolones that are widely used. It's highly active against a series of pathogens, and there's actually some MTB activity, which I'll talk about for this compound. So we thought it would be a good starting point to see if we could come up with something that would be a better MTB compound. So I'll go back a little bit of the history of this compound. The original hit was discovered by phenotypic screening at Pharmacia Upjohn. It's a compound called QPT1. It showed good activity against gram-positive bacteria. It was shown to have a DNA gyrase mode of action without any cross-resistance. Activity is in the minus isomer. Here's the absolute configuration of the minus isomer. It has 3 chiral centers in it. And Pharmacia Upjohn got bought out by the Pfizer Corporation. They had a robust optimization program and never could come up with something that could go into the clinic that had sufficient safety. So I had worked at AstraZeneca and led this project that led to zoliflodacin, that this compound shown right here. And we have I'm giving you just kind of a brief snapshot, kind of a Goldilocks and the 3 bears scenario. This first compound was an early, if you will, clinical candidate possibility. It showed modest activity, but was a relatively safe compound. But I think we wanted to see better activity against a broader spectrum of gram-positive agents, even though this was being targeted to be an oral MRSA agent. This is methicillin-resistant Staph aureus agent. The next generation of the compound of the series is this one right here with the triazole. This had better activity and the spectrum of activity that was envisioned for as a gram-positive agent. However, it had toxicity issues, in particular genotoxicity, and it was also toxic to pre-erythrocyte cell lines. So it would lead to anemia on had the potential to lead to anemia in humans on repeat dosing. And then the one that solved all the problems, both in having quite the spectrum of activity and being very, very safe, is zoliflodacin, and that's a compound in phase 3 clinical trials. So I'm showing this composite X-ray crystal structure of where these compounds bind. Now, I mentioned the DNA gyrase is an A2B2 tetramer. So there are 2 A units, 2 B units, 2 ATP binding sites, and these are where novobiocin binds and other ATP competitive inhibitors. And if we hone in at the site where all the action happens, I show that to the right. This is the binding site for the naptheridones, the compound from GSK. And here's a binding site for both SPTs and fluoroquinolones. The fluoroquinolone is in purple. And I show here in this a purple dot here, which is magnesium, which is bound to the SPT and otherwise associated with the enzyme. There is no metal associated with the cyan compound or the blue compound, which is the SPT compound. And I'll have more to say about that in a subsequent slide that differentiates these two compounds. So this crystal structure, of an SPT bound to the Staph aureus isozyme, was solved by folks at GlaxoSmithKline, GlaxoSmithKline, SKF. And that's a crystal structure with QPT1, the very initial hit that was discovered by Pharmacia Upjohn. So the modelers within H3D, this is Joe Ironman and Steve Feinberg, were able to derive an SPT binding model to MTB. There's crystal structures of MTB with a fluoroquinolone, but not one with an SPT compound. So here's their model with QPT1 bound. So within that, using this model, we could carry out a structurally enabled program for discovery or for optimization of SPT molecules for DNA gyrase. So in a nutshell, the analog design said, keep this black area constant. It doesn't look like there's room for modification. This area over here binds to GRB segment of the protein. This area over here stacks with DNA. There's considerable leeway of this R group over here to put in a large variety of substituents, and this interacts with another portion of the GRB bindings of the GRB protein and binding towards the GIA part of the protein. This is a clinolin resistant region of the protein. These are where residues that infer resistance to fluoroquinolones are found. There's narrow leeway for modification. So this is a compound of many that was ultimately came up with that is highly interesting. It has good MICs against MTB, and it's a good inhibitor of MTB gyrase. So there's a slight modification, if you will, in this southeast part of the molecule, and if you go over here to the northwest, a much larger modification. And here's a binding model for this compound that has been derived. So this compound was shown outright to be an inhibitor of DNA gyrase in cell-based assays, similar to actually a little bit more active than moxifloxacin as an MTB gyrase inhibitor. So here's a list of compounds, inhibitory potencies against DNA gyrase. These compounds are also inhibitor of Staph aureus gyrase, and I show a few numbers of data that was collected. However, I want you to look at these compounds in particular that were derived at H3D. These were earlier generation of compounds that AstraZeneca had found. These compounds showed some level of host cell MTB activity, and they also show activity against staph. The nice thing about these compounds that were developed at H3D is they got good MTB activity, but no staph activity. And there's a couple advantages of being selective for MTB over staph. And I should mention, it's not just staph activity. They were not active against a broad series of gram-negative pathogens, and they would be quite selective for MTB over over other pathogens. The advantage of this being that we have a lot of microbes in our gut, and we prefer not to knock them down with our antibiotic regimen. If that's one of the problems of moxifloxacin, it is a broader spectrum antibiotic, and it does hit not only staph, but can affect the gut microflora. So you'd prefer not to do that in a drug. Secondly, you'd prefer, if a drug came to the clinic, not be hitting other pathogens, namely because the series does have value, for example, treating staph infections. And if you're working out a dosing regimen that might be appropriate for MTB, it may be suboptimal for treatment of staph infections. If that's the case, you may be engineering resistance in trying to treat TB to staph if there happened to be a co-infection with staph or some other organism that these would have promise for. And that's something you don't want to do, because you want to leave it open for other compounds to be used for staph infections and not have resistance development in the other pathogens. Now, this is a compound that I had mentioned in the previous slides. Exquisite selectivity between MTB and staph activity, and a host of other pathogens. Now, what's the reason for this? We showed there's a fairly good correlation between enzyme inhibitory potency, MTB gyrase inhibitory potency, and whole cell activity that you see in this series. You don't get this correlation for other pathogens, for example, in E. coli and staph. So the observation that we made is, here's a plot of MICs versus log D, that for MTB, compounds that are more polar can get pretty good MICs against MTB. But more polar compounds are not populating over here. It's a more lipophilic compounds that get better activity against staph and against E. coli. So we think it's lipophilicity that's playing a role in order for the compounds to get into the cell. The long and short of it is compounds that are a little bit more polar, bacterial permeability matter less for MTB than they do for the other pathogens, in this case, E. coli and staph. This can involve passive permeability or active efflux by efflux pumps that are part and parcel of these pathogens. So it's not target potency, it's the permeability that gives us the selectivity. So this class of compounds was shown to induce an SOS response in MTB. There's another bioluminescence reporter assay that was developed at UCT that indicates DNA damage. So here's a cell wall biosynthesis inhibitor, isoniazid. It doesn't light up in the assay. Moxifloxacin does light up. If you get higher concentrations, it's two-sidal, so that's why at higher concentrations you don't see the high signal. And SPTs also light up in this assay. The SOS response is a hyper sensitive response that bacteria do to DNA damage. They're trying to repair that DNA damage. Sometimes things go off kilter and it actually ends up leading to cell death. So these compounds, like fluoroquinolones, are cidal as a result of this hypersensitive response. The Snappinger lab showed with a hypomorph that they had to gyrate to MTB gyre A, that they operate on target by inhibiting DNA gyrase. So this strain that was developed is hypersensitive to SPTs, as it is hypersensitive to moxifloxacin. So we're pretty confident that antibacterial activity you're seeing is due to the DNA gyrase inhibition. And in a collaboration with the Osheroff lab at Vanderbilt University, it was shown very nicely that SPTs bind to DNA gyrase cleavage complex of DNA. So in other words, DNA is doubly cleaved by DNA gyrase, and this transient state of double cleavage is where SPTs bind. So this is kind of shown in this cartoon that if you can imagine two cleavage sites in DNA, they happen to be 4 base pairs apart, and the enzyme otherwise surrounding the DNA cleavage complex. This is where SPTs bind. They can bind here and they will bind there. Similarly, fluoroquinolones bind here and they bind here. This is just a cartoon showing both SPT and fluoroquinolone binding at these cleavage sites. So if you cleave both strands of DNA and you get in a plasmid, you end up getting linear DNA. So this is circular DNA, and now it's linear DNA, and this can be detected in a gel. And you end up getting accumulation of this linear DNA in the presence of SPTs. And in fact, at the enzyme level for generating this compound 5867 was a little bit more active than moxifloxacin. And here's some of the activities that you will see in generating this linear DNA strand in the presence of various inhibitors. And again, including moxifloxacin was the compound in blue. So as a result, but despite the fact that these work similarly to fluoroquinolones, they are not cross-resistant to fluoroquinolones, as I mentioned earlier. So here's moxifloxacin, a couple of strains in which it is less susceptible than a wild-type strain. SPTs are perfectly active against mutant strains of MTB, and they're also perfectly active against other resistant strains to other drugs. For example, drugs that are resistant to isoniazid, still active, and drugs that are resistant to rifampicin, still active. And this is manifested not only at the whole cell level, but at the enzyme level. And again, this is work that was carried out in the Osheroff lab. The wild type enzyme, I'm showing you ciprofloxacin, moxifloxacin, and an SPT compound. You have similar activity. These are two enzymes from resistant strains. In particular, ciprofloxacin activity is knocked down considerably against these resistant isozymes, moxifloxacin a little bit against the A90B mutant strain, and considerably more against the D94G strain. However, the SBT compound is perfectly active. In fact, it's more active against these resistant strains that are out there. And that could be significant when you get to the clinic, that you could have a compound that would hit the problematic resistant strains of MTB a little bit harder. So that's good. And I mentioned they're not cross-resistant to fluoroquinolone-resistant strains. And as I had mentioned before, it has to do with magnesium binding. If you look at an X-ray crystal structure and otherwise do biochemical work, you can show that there is a requirement for magnesium for fluoroquinolone binding to SBTs. So that, in fact, the inhibitor is not a fluoroquinolone, it's a fluoroquinolone magnesium complex. But there's no need for magnesium binding of SBTs through that. And X-ray crystallography doesn't show any presence of a magnesium ion associated with SBTs. Now, this magnesium is otherwise bound to parts of the protein that when mutated infer fluoroquinolone resistance, but they don't infer resistance to the SBT. So with this, we have a novel mode of inhibition of SBTs and no cross resistance for the series. And finally, in the last science slide, there was a compound out of the SBT series that went into a whole animal model. There is a modest reduction of lung CFUs, a little bit more reduction of spleen CFUs. They don't work well as mainline MTB agents, for example, ethambutol. But it is promising that you're able to take. It is promising that you're able to get animal efficacy. So further optimization of this class is necessary if you want to come up with an MTB drug outright. So in summary, I've given 3 examples of target, if you will, repurposing and the idea of new antibacterials in RNAP. It's a structurally enabled project enzyme. It's a large enzyme complex, multiple ways in order to inhibit this enzyme. A lot of research work is going into that. For HAD-AB, the pyrazole acyl-sulfonamides were potent inhibitors, potent MTB activity without cross-resistance, another structurally enabled project. SPTs, structurally enabled, again, potent inhibitors without cross-resistance. Importantly, there's selectivity for MTB activity versus non-mycobacterial non-mycobacterial bacteria, and we're able to confirm the mode of action and the mechanism of action of SPTs. So here's an acknowledgement side. There's a large group of people working at H3D and a large set of collaborators that have worked on that. It's a little bit unfortunate. It makes it seem like the collaborators did the bulk of the work, but in fact, the bulk of the work was done at H3D. Several of these people worked on multiple projects that I had on perhaps all 3 of the projects that I described. And oftentimes people have small contributions, but you want to acknowledge what they had done. So a lot of good work done in this area, and that's what I have to present. And I want to thank you for your